With that, let's welcome our speaker today, Dr. Fergal Ryan. So Fergal leads the computational multi-omics group at Flinders University, where he explores how different types of biological data can be combined to better understand human health and disease. His research focuses on complex and poorly understood conditions, with a particular interest in how the gut microbiome and immune system interact with the brain and other organs. He has studied microbiomes from premature neonates to the elderly, leading major studies published in the world's top scientific journals. So without further ado, over to you, Fergal.
Thank you very much, Jake. I'll also begin by echoing your acknowledgement of Ghana country here and the people, and pay my respects to them. First as well, a slight apology. I was mentioning to Jake, I'm just getting over a bout of laryngitis. I couldn't speak at all on Monday. I was making a lot of jokes about the Little Mermaid and selling my voice, but I got over a bit on Tuesday and I'm feeling much better today. So I am actually here and able to give my talk, which is good. Over the last month, we've had some really fantastic talks that, like Jake mentioned, touching on the environment, gut microbiome, and kind of our relationship with them. And in terms of um, there's a real strong body of work now on that. We know that the gut microbiome is shaped by the environment around us. This is kind of established without any doubt. And we know that our brain is also influenced by the microbiome, but drawing the connections between all of these things can be very, very hard. But this evening, I wanted to kind of, I guess, tackle this from a slightly different lens. And I'm going to really start talking about some of the methods and kind of technologies that underlie what we do. Um, and touch a little bit on some of the history of them and how integral they are to the microbiome field. And then kind of at the end, touch a little bit on what I think are some of the really, really exciting things that are going to come in this field as well. Um, and so my background, you might not be able to tell, I'm Irish. I grew up in Ireland and did most of my early training there. And then I relocated to South Australia about 8 years ago now. Um, and so while I was in Ireland, I started my career in microbiology and very quickly realized that while I loved, studying bugs and kind of the miniaturized things of our world, I was really terrible in the lab. And so when I was kind of looking at next steps in my career, I think that one of the cheapest master's programs I could find was in something called bioinformatics. So I did it, and as it turned out, I was pretty good at it and kind of led into a whole career for me. And I got into this at actually quite a lucky time. So, one of the best pieces of career advice I can give people is always, well, get lucky, get into a field that's exploding right as you do. Um, and so now kind of cutting edge science is increasingly looking more and more basically data driven. So, if you go onto a TV or if you're watching a TV show and you're seeing them doing science and something like CSI or some medical drama, you'll often see something like this. It's got the multicolored test tubes. There's different sorts of vials and liquids around the lab. There's these sort of readout or real-time data being generated on a computer. Really ridiculous mood lighting. No one could actually work under these conditions. And several staff members will have mysterious pasts. But in reality, this isn't quite the case. And actually, kind of what a cutting edge scientific laboratory where someone has that eureka moment looks like now is probably something more like this. An office with computers. You probably can't see it here, but there's a lot of coffee cups around. There are lots of laboratories, of course. This is actually my office. I sit right here. Um, and there's the building itself has a lot of really fantastic state-of-the-art labs as well. But most of those labs are ultimately in kind of directed towards generating data that we then bring to the computer, which is where we, do that analysis, do that interpretation, and not so much at the lab bench and certainly not kind of sitting doing that next to your computer. And this isn't a new thing. This has been happening for the past couple of decades. So you can find a lot about this online if you look. This is just one article from the World Economic Forum that biology and data science are on a collision course. And I kind of think they've already collided, actually. And if you really want to be at the forefront of medical research these days, you need to have those data skills. And I think in the coming decades, actually having the ability to manage the data is going to be as important, if not more important, than knowing the biology itself. Um, and there are a few historical moments that have led up to that. You can go back to the 70s, which was the first time we got DNA sequencing. There's the advent of PCR, which with a Nobel Prize was awarded for that. But I think one of the really big ones was June 27th, 2000. So this was basically when the announcement came that the genome was cracked. This is a bit of a misnomer, really, but there was two basically kind of rival organizations that were racing to sequence or complete the full kind of composition of the human genome. One led by Francis Collins, who was leading the publicly funded Human Genome Project, and the other by Craig Venter at Celera Genomics, and that's both of them there. Um, and so this at the time was obviously it's rare that a scientific discovery is sort of leading the papers in this way. But this was viewed at the time as an absolutely enormous deal. You can go and find world leaders, presidents, prime ministers talking about it at the time, and how it's set to really revolutionize kind of our understanding of medicine and human health. And I think at the time, a lot of people, there was a little bit of a feeling of it was maybe the end of science or the end of kind of genetics, that this is it, we've got it, it's going to be easy from here on out. But really, this was kind of, now we look back at this as the beginning of a new type of medicine and a new way of doing research. When I was pulling up that last piece from the from the New York Times archive, I also found this editorial the day after, which I found was very, very interesting. It might be a little hard to read there, but just down here at the end. Nevertheless, having the entire genome laid out on computer databases will be a great advantage because researchers can search for relevant genes at speeds unimagined in the recent past. At Monday ceremonies, key scientists in the genome work recounted how genes that took a decade of hard work to find in the 1980s could be found today by a single postgraduate student in anywhere from 15 seconds to 2 weeks. So it's not just new questions that we can ask, it's the speed at which we can ask old questions. And this actually hasn't slowed down. If anything, it's actually sped up since then. When we think back to what the human genome looked like back then, they used to always have this printed out. And so here's each of the chromosomes, and you can go in and read them. It's literally just strands of A's, T's, G's, and C's over and over and over again, right? There's actually, if you're interested, there's a very, very funny clip from like a medical drama show where, they're trying to figure out what's wrong with the patient, and they sequence the genome and they think it's a genetic disease. And the head doctor is asked, are they going to analyze this with software? And the doctor's like, no, we're going to go through it by hand. And in the next scene, it's them literally reading through the genome by hand, which is absolutely ridiculous, by the way. It would absolutely never happen that way. But people sometimes think that's what you might do with it. Um, so what it looks like when you're actually generating this data back then with something like this, and there was a lot of rooms looking like this, where you'd have computers reading out from these machines that are called Sanger sequencers. It's very accurate, but very slow. Jump forward to today, many research institutes would have a suite like this. And you'll find one of these, I would probably say, in every major city in the world. This is a machine by a company called MGI. There's an Illumina machine over there. And you can see these are quite big, but each of these machines will do kind of what it previously would have taken multiple rooms of this size to do. We also now have machines like this, handheld DNA sequencers. They don't produce as much data or as much sequence as some of these bigger ones do, but they mean handheld means you can take them out into the field. And as we've had all of these technology developments in sequencing, what that's meant is the cost has dropped spectacularly. So the original human genome, the first one, cost adjusted for inflation, something like 5 billion US dollars. You can now generate that same genome for about 100 US dollars. So we've seen a drop from over like in the billions to the hundreds. And what that has meant is that these technologies have really now kind of spread everywhere. So, a lot of people will have heard of companies like 23andMe or Ancestry.com that will use those for ancestry-based things. But there's lots of other areas too. So I think during the COVID pandemic, we all would have seen the maps where they were tracking cases. that was one of the first times actually that DNA sequencing technologies was deployed rapidly in real time to monitor and control and track a disease outbreak, particularly in Australia. But it's used in other contexts as well. So I have some friends working in pathology that will use it for looking at origins of stuff like foodborne illness, and they can track it back to the exact food and identify where it's from. You can also use it for looking at stuff like antibiotic resistance in quote unquote hospital superbugs. So there's that. There's stuff like forensic genomics. The Golden State Killer case was the famous example from that. There's ancient DNA studies, which to this day still blow my mind that they can get DNA from stuff like Neanderthals or look at human migration patterns going back thousands of years. And then there's obviously a big world around this in terms of agriculture and food security as well that we've heard more about this month as well. But for me, the really exciting thing that came from this actually isn't the DNA, it's everything else that came from it. So DNA is the molecule that kind of has the blueprint for what our cells can do. And so every cell in your body will have the same DNA, but those cells all do different things. So a skin cell and an eye cell and a tongue cell all have different functions, but they have the same blueprint underneath, but the parts that they actually use to build it are different. And this is where other molecules in the cells come in, stuff like RNA and proteins and metabolites and lipids. But actually, through the development of technologies for DNA sequencing, we also built technologies for measuring all of these. And collectively, these are referred to as what's called omics technologies. And so what this means is that for someone like me, it's now pretty routine to generate something like 100,000 data points from one biosample. So in a single study, we might, for example, we had a study published earlier this year looking at the gut microbiome and immune development in babies kind of over the first months of life. And for each baby, we had multiple samples. So that was that number of data points times the number of babies times the number of time points. The data gets, really, really big. But of course, the absolutely incredible thing about this is that we don't need to go in there with a hypothesis about one protein or one gene. We can actually just measure everything. We can just measure all cells in the immune system, all bacteria, all molecules simultaneously, and simply see, well, what's wrong? And then we can just from there adjust accordingly. So a really nice example of that was, there's lots and lots of examples from that. I just always go back to this study. So this is from a group out of Harvard who are huge in the microbiome space, led by Professor Curtis Hottenhauer. So this is an example of a drug called 5-ASA, which is used for treating inflammatory bowel disease. So inflammatory bowel disease is a very well-established disease known to be impacted by the gut microbiome, and 5-ASA is a good treatment for it. But 5-ASA is quite variable efficacy in treating IBD. Here, what this team did was they combined a bunch of these omics technologies to actually identify there's a specific microbe responsible for this variable efficacy. And actually, it's a specific enzyme within that specific microbe that's responsible. And so what this means is that they can now basically develop a test to see, well, if you have this microbe, maybe you need an antibiotic or diet intervention to adjust your microbiome for that. And that will help improve the efficacy of something like 5-ASA, which hopefully will keep your IBD from going into flare. IBD is a very, very tough disease for those that are living with it and managing it. So these types of things are extremely important if we're able to make medicines work better, basically by using this. But dealing with all of this data is very, very tricky. So as I was talking about, you had that 100,000 data points times the number of patients, times the number of samples that you've collected. To give you an idea of the scale of this. So we often, if you think about computers, you'll go into a shop and you'll probably see words like gigabytes and terabytes in terms of how much memory they'd have. And so a high-end laptop today would have probably a terabyte of storage. In total, you can store a terabyte on there. And that would be an absolute ton of photos, a bunch of videos, all your Word files, PDFs, everything you can imagine will fit onto that for the most part. When you jump up a kind of level, you'll have what's called an exabyte. An exabyte is 1,000 terabytes. So, for example, all global YouTube video uploads per year are about 2 exabytes per year. So 2,000 terabytes per year of just video uploads. Just this year alone, omics data is projected to produce over 40 exabytes. So basically, it's producing more data than essentially everything else in the world combined, which is pretty insane when you think about that, the level of it that's required. It's good for people like me because it keeps us in work. It means there's actually far more data than there are people to analyze it, which is nice for job security, but it's bad for actually making progress in science, which is part of the reason I'm always keen to interact with groups like this and audiences like this to really kind of communicate to people, this is a field where we're desperately in need of people to help. Um, so when you have like a small data set, if you went back to what we might have been doing in a microbiome study 10 or 20, or not even 10 years ago, 20 or 30 years ago, you might have something like this, comparing the amount of E. coli in a disease group versus a control group. If anyone's ever studied statistics, you've heard of the student's t-test. And you can use this to compare the abundance of E. coli in your two different groups. It'll give you a p-value, and you'll be able to say whether or not it's a difference between the groups. A little fun fact, the t-test actually came out of the Guinness Brewery in Dublin by a guy called William Gossett. It's called the student's t-test because he published it under a pseudonym, student. We always learn about this in university or high school, and so people often think it's students for students that are learning it, but no, it's a pseudonym. I always enjoy that little factoid. But when you scale that up, imagine you were measuring the abundance of 10,000 different bacteria between groups of 500 people. Suddenly, these tests get very difficult. Maybe if you're doing it in one test, you could probably actually work it out by hand, or maybe even with a calculator, right? When you start going up to thousands and thousands and thousands, you need to start automating these processes. You need to start applying more advanced techniques because actually you end up with these weird artifacts and signals coming through when you measure that many different things. And so that means we need what's called a cross-disciplinary approach. So where we can take someone that has expertise in biology and medicine, and then they're able to use stuff from computers and data science to programmatically and automate statistical tests to be able to answer questions, okay? And now you're probably sitting there, we're 16 minutes into this talk, and you're saying, Fergal, you've barely talked about the microbiome. This is Soil Yourself September. Please, can we go to the microbiome? And I'll say, okay, we'll get to the microbiome. So this is the number of scientific publications published per year containing either the word microbiome or microbiota. The reason I spent all of that time telling you about data and cross-disciplinary and DNA sequencing, it's basically because this spike here in microbiome research, this is a spike in our understanding of the microbiome field, actually. What it represents is new knowledge regarding to the microbiome. This happened basically because of DNA sequencing. So the human genome was completed around here. This scientific paper by Ekberg et al., published in 2005, is really one of the seminal microbiome studies that we would recognize today as modern microbiome science, where they, for the first time, were able to look at bacteria in the gut and identify, oh, wow, there's a lot of these. We should probably be looking at these more. And so back here, it was kind of, in the tens of papers, into the dozens of papers, maybe starting to get into the hundreds. And now we're looking at on the orders of, 20 to 30,000 per year. And it's really because growing these bacteria in the lab is extremely difficult. In many cases, we still even can't do it, and we know what these bacteria are. But DNA sequencing means we don't have to grow them. We can just measure them directly in human samples. And it was this breakthrough is what told us that the microbiome is associated with all of these different diseases, whether it's, gastrointestinal infections, through different cancers, through the efficacy of drugs, to obesity, to everything, right? And so this has really been extremely important. But because DNA sequencing is data dependent, microbiome is data dependent, okay? And so these things all naturally lead on to each other in a way that I think the people that were running the Human Genome Project 25, 30, 40 years ago would never have even considered that, the technologies that are developing there will end up leading to something like this, which is also one of the really important reasons that, are, for government and public bodies, that we actually fund these sort of big blue sky endeavors where we actually just look to solve the big problem, and don't, I guess, always try to have stuff so targeted. Because, if we had set out to cure cancer, we maybe won't get there. But whereas, when we try to solve a big problem, we find these unexpected things like this. If you're interested in learning a little bit more about the history of the kind of microbiome field, there is this a a site called Nature, which publishes one of the really major scientific articles, has developed this multimedia timeline of human microbiome research that goes right back from the field being born and early stuff in the 1940s right through to the modern day. You can get it from this QR code, which I can go back to if you want at the end of the presentation. Or else you can get it as well from there. But one of the things I wanted to show from this is across the full history of the whole microbiome field, one of the things they identify as one of the major milestones of the field is the development of a piece of software called QIIME. It's spelled QIIME. A lot of people call it QIME, but it's pronounced QIIME. And then this basically allowed for standardized analyses of microbiome data across different studies. And this was just an absolutely enormous breakthrough, and it's been used by tens of thousands of scientists around the world to analyze their microbiome data, which has really facilitated, again, and enabled us discovering these patterns that lead to what we know about kind of the microbiome and human health today. And so now you might be saying, well, Fergal, what about the gut-brain axis? So right around the time, just after the human genome was completed, and right before Eckberg et al. Published a seminal paper on microbiome science, there was another group of people, a group of authors called Pseudo et al., excuse me, that published a paper that showed that mice without a microbiome have heightened stress responses. And so this paper is often recognized now as kind of the beginning of like the modern field of gut-brain axis research. And so much of this work happened in germ-free mice. And these are mice that are basically where we've removed their microbiome. They're all genetically identical mice. Many of them have a genotype called C57 Black 6, one of the more common genotypes of mice that we use. And of course, one of the useful things about this is that we can see what is different about these mice, that they have heightened stress responses, or there's tests that they do where they see that these mice are less social when they're kind of put into different groups. But actually, what that means is we can put a microbiota back in these mice and see then that we can make the mice more social or actually help with that stress response, which means that we can show that the microbiome isn't just associated with a change in the brain, but it can actually reverse it as well, which is extremely, extremely important. But of course, while something like germ-free mice allows us to show that the gut-brain axis exists, shows us that it's real, something like omics technologies allows us to ask how that works. And so this was much of my early work in the field, actually, just when I was coming up early on in my PhD. I ended up back in Ireland doing some work with Professor John Cryan, who's a neuroscientist and an absolute kind of titan of the gut-brain axis field, where they were doing this, using those sequencing technologies I talked about earlier on brain tissue from these mice to be able to identify what is it about it that have changed. And from this, we were basically able to really dig deep into the molecular mechanisms underpinning this. And this type of work has been extremely important because whereas measuring something like how social or stressed a mouse is, they have standardized tests for these, but they're always a little subjective, of course. Something like gene expression is far more standardized and quantifiable as well. So we've been able to show that this is a real quantifiable effect, and we're able to show very strong scientific and molecular evidence for that. And so that's really where omics technologies have so far, I think, been an absolute pinnacle of importance for the gut-brain axis field. I've only shown you some of my papers here. There's far better people out there that have done this in a much, much better way than I have, but I'm a little self-centered when I was making the slides, so I just picked some of my examples. But of course, we still have a long, long way to go. In science and medicine, we have a habit of keeping our disciplines separate. And this is probably why something like the gut-brain axis actually went undiscovered for a very long time. Neuroscientists and microbiologists, they're based in different buildings. They go to different conferences. They publish in different journals. They aren't always interacting, perhaps in a way that we would like them to. I think to kind of break down those barriers. And I think this is why so many now of our chronic diseases that are linked to the microbiome are diseases where they do span these different disciplines. And it's going to be extremely important, I think, to cross between those to really kind of understand them and ultimately develop treatments for them. A really nice example from this I pulled out earlier this year. I was reading an outlook in Nature. And so Nature, again, is one of the really top scientific journals. And this was from someone talking about working in the Alzheimer's field. um, going back a few decades ago. And so this was right around the time earlier this year, there was basically a bunch of high profile failures in Alzheimer's drug clinical trials, where for obvious reasons, this is a really, really difficult disease, and we want to develop effective treatments for it. But a lot of the treatments have focused around this thing called amyloid beta. But actually, a lot of the drugs that have been targeting those pathways, they've not been working. They're safe in humans and they haven't been causing off-target effects, but they're not treating the symptoms. This section from this outlook I found interesting. Historically, the Alzheimer's field has been less than welcoming to theories that do not center around amyloid beta and tau, the proteins that accumulate to form plaques and tangles that are the hallmarks of the disease. Henneke, who's the name of a scientist or profiling in this piece, encountered this himself while exploring the potential contribution of inflammatory processes in the late 1990s. And to quote him, you were just lucky when you weren't beaten up by the amyloid beta or tau people if you would mention immunology. I think now coming on, 30 years or so from there, we know that actually all of these systems in our body are a lot more connected than we anticipated. So actually, when it comes to looking at these diseases, we need a microbiologist, we need a neuroscience, we need an immunologist. We need them all to be talking to each other, and we need them actually in the same room. And we probably actually need people that know a bit of everything, that are going to be able to put those pieces together. And to kind of bring that full circle, that's where I think the data is going to be so, so, so important, because stuff like our omics data, it allows us to just look at everything. We're not just measuring immune cells. We're not just measuring stuff from neurons or stuff in the brain. We really can look at everything. And I think that's going to be important for breaking down some of those walls between disciplines. Because we don't kind of filter a priori for what we want. We take this broad approach where we'll just do everything. For example, me and my colleagues right now, we're in the process of putting together a study doing this, where we're looking at something called chemotherapy-induced cognitive impairment. So this is something that happens to 30 to 40% of people that are undergoing chemotherapy, where they'll develop a kind of a brain fog, or sometimes called chemo brain. And so we're using these technologies to understand if A, can we predict who's going to develop it before they start their chemo? And B, is there anything that we can do to treat it? Perhaps is there already some safe therapeutic that we can maybe look at repurpose to help with that? But the approach we're taking to that is just to measure everything. We're also looking to combine these technologies with neuroimaging, stuff like more, I guess, traditional neuroscience or neurological approaches, like MRI, where there's a real exciting emerging field now where we're going to start seeing research studies combining these molecular technologies, measuring all these molecules with neuroimaging data, something that a neuroscientist or a neurosurgeon is going to be a little bit more familiar with. And I think that is going to be an absolute game changer for the gut-brain axis field, particularly if we're able to start doing that at scale. I think it's going to be really, 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 really amazing. Something else I'm really excited about, not just in the gut-brain axis field, in the microbiome field in general, is this idea of ingestible sensors. So I did my PhD in a microbiome research institute, which I started, oh God, almost 15 years ago now. And back then, of course, we all kind of started out being a little bit grossed out by, fecal samples and poop. Over years, you just get used to it. It was someone might be going into the lab to culture bacteria. Guys, I need a fecal sample. Who needs to go? Okay, there you go. Take the pack, off you go. Bring it back into the lab. And you do kind of get used to that, but obviously that isn't always the case for everyone. And so there are kind of some barriers around that and working with fecal samples. One of the problems with fecal samples, though, is that fecal samples come from basically the end of the gastrointestinal tract. And our gastrointestinal tract isn't just one thing. It's actually very different throughout. We start with the stomach, into the small intestine, and then the large intestine, right? And actually, the bacteria along these different parts are very, very different. The amount of bacteria is very different between them. The amount of oxygen is very different between them. You actually have a lot more oxygen up here in your stomach and a lot less oxygen down here in your large intestine. And then even just the kind of cells are different and the different types of molecules are different between them, right? And so what this means is that when we measure a fecal sample, we're probably mostly getting just what's at the end here. We really can't measure what's happening up here. And this is also a barrier actually for studying the microbiome. But just in the past couple of years, we're starting to see the developments of these basically little capsules that you can swallow, that will have different pH sensors that will break at certain parts of the intestine. So they'll take up bacteria and metabolites and RNA and stuff like that at each point. You'll then will excrete them out into the feces, and then we can test those. So we're able to, in a non-invasive way, study each of the different parts of this. My PhD was in inflammatory bowel disease. And so we were really interested in looking at the bacteria that adhered in the colon down here. And so the study that we ended up doing was that we were looking at patients who were coming in for a colonoscopy, and the gastroenterologist that we were working with would end up taking a mucosal pinch biopsy from here that we obviously the patient also had consented to this, and that we could then use for our sequencing approaches. But doing that obviously is hard, because the only way you can actually sample that, a patient already needs to be coming into the hospital, which makes it very, very difficult to actually get a representative sample of just healthy humans. And what is the healthy human mucosa, right? It's hard to do that. Technologies like this allow us to get that look inside the body, which again is going to just be an absolute game changer for the field. There's numerous companies now that are working with it and developing products around this. So this is really, really going to be big in the years to come. Aside from that, kind of just some other thoughts on what the future holds before I kind of wrap up. So as I've kind of been talking about through this, over the past 2 decades, we've seen a lot in laboratory models, and particularly germ-free mice. And this has been essential, really, in providing the foundation for identifying mechanisms. And this has really provided the proof that gut microbes can influence brain function. So I work a lot with clinicians, and particularly if you were to talk to an emergency physician, if you were to talk to, doctors actually across a lot of different specialties, they probably still don't quite believe this, actually. But we know it's true in these different animal models, and we've been seeing really interesting results with fecal microbiome transplant in different diseases, whether there's some interesting results across Parkinson's, and some very interesting results coming out of bipolar disorder as well around this. Um, but actually, people still don't quite believe it, I think, when it's just in these laboratory models. So I do think the next phase, we're really going to have to move into humans. And I think these omics technologies and stuff like those ingestible capsules, or combining it with technologies like MRI, is going to really, really, really be central to that, to developing. What I think we need in humans is a robust, quantifiable readout of gut-brain axis, something that we can measure in humans on a large scale. I have a few ideas of how we might do this, and it's something that me and my research team are looking at doing kind of in the future, where we can really apply this in large clinical cohorts. Because if we do this, this doesn't just allow us to look at diseases where we suspect there's gut involvement, it allows us to look at all diseases and just see that again in an unbiased way. That idea of simply measure everything. The other thing I'm really, really keen that I think we need to start looking at when it comes to the gut-brain axis, we think a lot about, does it exist and how it works. Something I see very few scientists talk about is why. Why did we evolve this system? It's conserved across different mammals, which implies it provided some sort of competitive advantage. Earlier, I forget, was it last week or the week before, when Jake was speaking, he mentioned the old friend hypothesis, this idea that we co-evolved with microbes. You can see very easily, or you can imagine a lot of different reasons we would do this for our immune systems. But why the central nervous system? Why the brain? It's a little bit of an open question for me. I think it probably speaks to the fact that actually the nervous system, the immune system, the digestive system, I think all of these are probably interconnected on a fundamental level. In a way, I think that, again, going to how we split up our disciplines, how we teach the stuff, and how we structure stuff like universities and hospital medical departments, kind of puts up these barriers between things that I think actually isn't the case in the body. But on that thought, I'm going to leave you and just say thanks for listening. So I'm a researcher at Flinders University. We've recently just had this fabulous new research institute built, which I spent a lot of today talking about how we don't need laboratories anymore, but we're very, very lucky to have some really incredible laboratory space in this building. I'm very, very lucky to have a lot of great colleagues there, and great students, and a really great team. So thank you very much, and I'll very happily take questions.
Thanks, Fergal. Round of applause. That was a great talk. Please add your questions in the chat and I'll read them out to Fergal. Let me try and think of some in the meantime. Um,
I'm just going to jump back to this timeline while I do. I think it's if
Go
you're
for it.
learning more about the microbiome, it's a really nice resource. It's particularly I'm such an absolute nerd for history. I normally have a history book next to me. I don't right now, but I absolutely love getting the history and the kind of larger context of the field.
Great, thanks. So, do you know which kind of clinical conditions, so depression or Parkinson's, etc., show the strongest, most reproducible gut-brain signatures?
I so this is something I have a PhD student at the moment writing a review on this,
Cool.
and it seems to be Parkinson's actually, interestingly. So, one of the things around Parkinson's disease is that there's some very interesting data from public health studies and epidemiology that shows that inflammatory bowel disease, which is a disease we really know is related to the microbiome, is actually a risk factor for developing Parkinson's, unless the IBD patients get an early treatment with certain anti-inflammatory therapies, which could then actually reduces the risk of developing Parkinson's either. So it implies that it's not just a kind of correlation, but there's actually a causation there. It's also in
Mm,
Parkinson's, a subset of patients will experience gastrointestinal this symptoms years before they ever develop motor symptoms. So it really hints to this kind of gut first disease, actually, which is very, very interesting.
mm,
Multiple sclerosis is another one I think probably has a large microbiome involvement, but that's probably because MS is kind of fundamentally an autoimmune disease, and the microbiome is a really important immune regulator as well. Bipolar, I think, has got some very, very interesting links to. I'm also interested in I think around dementia. I think there could be something there, but I think it might be a bit indirect. There's some interesting evidence recently that infections and different microbes are able to modulate the gut, or sorry, the blood-brain barrier, which is kind of what keeps our blood and or basically protects our brains from the environment. And so if you can have the wrong, I guess, bacteria in your gut, it makes that more permeable, which then can lead to stuff like developing inflammation in the brain that perhaps over the course of 20, 30, 40 years can lead to something like dementia.
interesting.
Um, yeah, so that the well, maybe they're just my crazy ideas for now, but you
Mm,
start to see these sort of correlations on large data sets that seem to hint at these patterns that
mm,
kind of do break down. We've known for a long time there's genetic components to these diseases, but there's also an environmental component that we just can't tease apart, and I think that's probably some in there somewhere.
cool, thanks. So they say that something like 80, it's a crosstalk, obviously, but there's something like 80% goes to the brain, 20% goes from the brain to the gut. Is that, in your experience, the kind of how you would view it?
I think so. Yeah,
Think
yeah.
so.
Yeah, I guess, but look, I'm coming at this, like I said earlier, I started in microbiology, so I come at all these problems from bugs first. I think.
Yeah.
So yeah, yeah, which again is the importance of having kind of people coming in from different disciplines, really kind of coming together to try to solve these problems.
Interesting. Yeah, because a lot of focus on looking after the gut-brain axis seems to be through the diet, and I'm always thinking, well, obviously it's a two-way system, so we should also be looking after our mental health to look after our guts
Uh,
as well.
Oh, 100%. Yeah. I mean, and
Yeah.
look, there was a really big study just published a few months ago in Nature Medicine, and it kind of reinforced. Again, it was using all of these big omics technologies on 50,000 people in the UK, and it found lots of interesting signals, which would be important for disease. But one of the
Mm,
top things that came out as predictive of health, exercise, social interaction, the things we know work, right?
yeah.
But I think when we're able to show that with a really cutting edge molecular test, maybe that's more impactful. I don't know.
Yeah. All right. John says, how far back in the evolutionary tree does the gut-brain axis go?
This is a really good question, and I don't think anyone knows the answer to that.
I suspect
Um,
it's hundreds of millions of years, though, right? Because
yeah,
don't some
yeah,
flatworms have some kind of primitive version of it?
yes, yeah. So I have a couple of ideas about how you could look at that from an evolutionary perspective. It'd be something, Jake, I'd be interested to talk to some of you guys about this. Something like if we go back and look at animals that have been under different infectious disease selection pressures, something like animals from the Arctic versus animals from like a more tropical environment.
Mm,
If we see a different kind of selection there, but it seems to be very, very old. And you can imagine my kind of current working idea is the basic thing we recognize now as the gut-brain axis started because as we were evolving a central nervous system, any infection there is absolutely catastrophic. So right from the get-go, our immune systems needed to be primed to act quick. Um, and I think we've just kind of evolved to use those signals in different ways.
Yeah.
But it's such a big open question. And I don't know about you, Jake, do you ever see anyone asking that in the literature? I never see it.
No, not really. But I mean, as I say, I think like the enteric nervous system at least is probably like 500 or 600 million years because there's evidence of it
Yeah,
in
yeah,
flatworms and stuff. And then I think the central nervous system's probably evolved from that when the vagus nerve appeared. But so I
yeah,
suspect it's hundreds of millions of years. But yeah, I don't really hear many people talk about it often.
yeah.
Any more questions? Please put them in the chat. So in the meantime, what are your main what are the main challenges that you find in this field?
Honestly, a really big one is actually recruiting people. So,
Okay.
like I said, so we've so much data and we've so many kind of big problems. But so if any of you know sort of people that are like 16, 17, 18, thinking about careers in science, come and
Yeah.
go and tell them there's an enormous amount of data that are just waiting to analyze. And recently we just keep having people tell us that, well, LLMs and AI is going to solve this. I use the AI tools every single day. I love them. They're amazing, and they're not going to
Yeah.
solve this. At best, it can kind of increase our productivity, and maybe, three people can do the work of 10, but it's not. It's kind of not really going to solve the problem either. So yeah, please just, that's always kind of the big barrier for us, is actually recruiting, getting into students, getting into kind of young scientists that are going to come up and be actually the next generation of leaders that will come through and hopefully surpass us all, right?
Hmm. Yeah. So that's a good point though. So how might AI and large language models, etc., help in generating testable hypotheses and just generally in bioinformatics moving forward?
Yeah, so I think they'll be useful, I think, for breaking down a little bit of the barriers between those disciplines, where
Yeah.
you probably can, as a neuroscientist going into microbiology or vice versa, ask a complex question and probably get a pretty reasonable answer. Sometimes the problem is, of course, these tools still hallucinate so much. We can't really
Mm.
trust them. The main thing I found for me is that and this is probably true of anyone if you're in a public sector job, and probably even a lot of us in the private sector. I spend an enormous amount of time writing reports. I can use AI to speed up the process of writing those reports, which means I can spend more time on the science. That's been
Yeah.
the benefit for me.
Yeah. Okay. The admin side.
Yes, basically. There's a question
Cool.
there, I think, from, is it Kay, is
Yeah.
it?
Craig's put one in the chat.
Oh, cool.
Left field question. There are electromagnetic field theories of consciousness, and different brainwave frequencies are linked to mood states. Will gut-brain science need to start looking at resonant frequencies of different metabolites and molecules, and potential changes, sorry, in bioelectric fields? It's a bit of a
I don't, yeah,
light
I don't know
question
much
for you.
about bioelectric. Yeah, I don't know much
Yeah,
about bioelectric fields, so I can't really comment on that.
yeah,
But again, in principle, this is the kind of thing with the big data approaches, right? The more
yeah.
things we measure, the more questions we can ask and answer. So if there's
Mm.
something there, we can hopefully assess it for sure.
Cool, thanks. Is it Kay or Kai? You've got your hand raised. Would you like to speak in person?
Yeah. Yes,
Go for it.
thank you.
Go for it.
Thanks. I'm really enjoying this series. I'm thankful that you've offered it where you have.
Oh, thanks.
Yes, Fergal, I'd like to ask you uh, how progressive you're being in regard to your recruitment. There are, of course, ways that you can incentivize the interest of study and work in the field you work, and similar by approaching individuals who've been left out of society, don't know what to do, want to help. And there's ways to, of course, locate them and deliver some sort of incentive. A few groups I can think of right away are, of course, people who are suffering, but not much is being done to help them. So, since there's a war on women and girls, people of African and Indigenous heritage globally, you could reach out to them. Same thing with coercive control, Machiavellianist, narcissistic abuse, etc. People en masse are going through these things, being ostracized, etc. So I would strongly recommend maybe linking what you do with, especially since you work in integral medicine, or you connect to so many different areas that do affect their lives. I think you'd have no problem whatsoever to recruiting droves of people if you go to the scapegoated, the most targeted for abuse, fraud, crime. If you go after those populations, I know for sure that if you connect it to what they're going through, show them what they benefit from, that they will be involved in this world that can propel so much positive change for our species and home and provider, the planet we share. I know for sure if you can show the link of what you do to what they go through and them and all the work you do benefiting them and the whole world, absolutely you will be successful. And thank you for offering this meeting.
No, thank you, and you're 100% right. I guess to clarify that that's not just, I guess, a problem for me and my recruitment. It's field wide and it's very well established. So in addition to my work at Flinders University, I'm also the vice president of the National Australian Bioinformatics Society, and so through that, we're, I guess, extremely cognizant of this fact. So bioinformatics, actually, we see a lot of really great examples in our field of female leadership. And one of the things we really like to point out to that, I guess, is versus some other areas of science, particularly when so much, I guess, of the burden of stuff like rearing children and taking care of homes actually ends up falling on women in society. It just does. Bioinformatics actually it provides a more flexible working lifestyle in some ways because so much of the work can be done remotely. Through that field, we also actually do quite a lot with indigenous populations. Not me personally, but one of the former president of our society actually is one of the leaders in the country in indigenous genomics and does a lot working with Professor Alex Brown, who's one of the real superstars of indigenous research here in South Australia. That we've again been very, very lucky to have that as part of our community. But you're absolutely right. For me, I think one of the barriers I often have is here at Flinders University. Many of the students I'm exposed to are students that they the career trajectory they're super keen on, and the way that they want to make a difference is via studying clinical medicine and going on to become doctors. And the thing I'm trying to kind of pry them away from is, come with us and look at these problems from a different way. And yeah, kind of taking that other approach. But I absolutely agree with you on those points. And there's things I'm trying to do, but also, like I said, plugged into the larger society to try and help with that.
Nice, thanks, Egil. Building on that, are there what kind of opportunities are available for citizen scientists to get involved in gut-brain axis and scaling that kind of research up?
That's a good question. It's probably something we should think about more through Grounded Minds, actually. It's so one thing that is extremely useful to scientists is when we go to governmental funding bodies, they want to see that we've consulted with the community. Through the language that the government always uses for this is consumers. I personally always find that language a bit odd, but that's the terminology that's always used for that. Whether or not that's something that we at Grounded Minds could actually look at bringing that together, which would actually find a way for a citizen scientist to get involved in a tangible way, get to meet people, and just get the conversation starting, which actually might lead to right to kind of building some of those other connections.